really excited to have Dr. Derya Unutmaz with us today to talk about the crossroad of the immune response and microbiome and the impact on ME CFS. Go to the next slide. So Dr. Unutmaz is a professor at the JAX Lab for Genomic Medicine in Connecticut and the director of the JAX Collaborative Research Center, which is funded by the NIH. He has developed various methods to better understand human T cells and is driven to understand the role of the immune system dysregulation in a variety of chronic diseases, including cancer, chronic fatigue syndrome, and aging. Dr. Unutmaz received his MD from Marmara University Medical School in Istanbul, Turkey, and completed doctoral studies at Novartis in Switzerland and NYU. So thank you, Dr. Unutmaz, for joining us today, and take it away.
Thank you very much, Sadie, for the kind introduction, and also the organizers, Emily and Allison. So very pleased to be here. So what I'm going to try to do is tell you a little bit about the work we're doing, trying to understand ME/CFS disease from a biological perspective. But before that, let me give you a little bit of background to myself. I trained as a physician, although I haven't seen patients for a very long time. I've been doing research for the past 25 years or so. And my research is exclusively focused on human immune system. Try to understand why the human immune system works the way it is, or why it doesn't when it's perturbed during conditions, chronic illnesses. We've studied HIV for many years, which attacks the immune system. And we also try to learn from the patients what's going on and try to decode that in the laboratory and understand the complexity. And I will get into that a little bit more. Guess, do I have the slide controls? Okay, I think I went too far. Should go back. Can we go back with the slides? I'm not sure I have that control here. Is it possible to take the control back and take the slides? I don't see the arrows to move the slides back forward. Oops. Okay. All right. So let me try this again. Sorry for that. All right. So let me give you a little bit of a background why I was interested in ME/CFS, and that led to this NIH-funded center last year. And as I said, I'm a human immunologist, and I've been studying different chronic illnesses, infections like HIV for many years. And I became interested in ME/CFS, I believe, around 2014, when actually I met Susan Vernon, who was at Solve CFS at the time. And she convinced me that this is really an immunological disease, that we should really look into it more carefully, and sent me some patient samples that she had stored at Solve CFS. And honestly, I wasn't really expecting anything profound to see. You know, the patient numbers were only 20 subjects, I think, at the time. But remarkably, we did see differences between the healthy controls and the patients, and that got me very interested in this condition. And soon after that, I also realized how neglected disease this is. You know, it's really a biological condition that in some circles wasn't even considered as such. So that also increased my interest. And about 3 years ago, I moved to JAX Laboratory. This was when I was at NYU School of Medicine. I was a professor there, and about 3 years ago, I moved to JAX Laboratory in Connecticut. We wanted to study this in much more detail and assembled a team. Suzanne, I think, moved around that time to Utah to work with Cindy Bateman, one of the leading physicians in the field of ME/CFS, in her clinic, and that enabled us to form a wonderful collaboration where we are currently recruiting our patients, and we hope to set up another cohort locally in Connecticut as well. And that eventually led to our application for this big center grant. We formed a really wonderful team that I will tell you a little bit more about at the end. That led to funding from NIH that we're very excited about. So let me just start with a very overall view of what we're trying to accomplish here, or what is our hypothesis, and then I'll tell you why we think that's important. So basically, we're obtaining clinical samples from patients that Cindy Bateman and Susanna Vernon recruits based on certain inclusion-exclusion criteria. And these are not just blood samples that we get, but also stool samples, because we're trying to analyze the immune system in a very high detail. I'll tell you more about that. In addition, we want to understand the microbiome being the mix or metabolism, these small metabolites that impact our immune cells. So the idea is that we want to approach this in a systems biology perspective. What that means is that we want to collect lots of biological data points, of course, based on certain hypotheses, and then create a computational interactome, which we can link to the disease states of these patients. One important thing about to remember, and this is a point that I'm going to try to make at the end, is that this is a very heterogeneous condition. In fact, most diseases, even very well-defined genetic diseases, are heterogeneous among patients. So it's very important that we try to link the biology to patients' clinical symptoms and their other factors such as the environment, disease duration, and so forth. So let me first start with why the immune system. Why should we care about that? Obviously, I'm biased to think that it's important, but I want to try to convince you that it really is important to study. So I think we all know that. Let's try to. Okay. We all know that the immune system is very important, very good for us, because without it we cannot function, we cannot survive. We need it to clear infections and repair tissue damage. Every time you have a flu infection or bacterial infection, it's your immune system that's trying to clear the infections and then repair the damage. So that's the good part. Unfortunately, there's also a bad part of the immune system, and that happens when the immune system recognizes, or rather does not recognize self as self, but thinks that is a foreign organism. For example, your pancreas is recognized as some sort of a enemy or a foreign object, and the immune cells start to attack, and that causes autoimmunity. We have many autoimmune diseases like multiple sclerosis, diabetes, type 1 diabetes, and so on. And that's the bad part. And there's another bad part, for example, allergies, and the immune system recognizes things that are not infectious, for example, derived from food like peanut allergies or pollens and things like that, that are not necessarily dangerous for us, but somehow they're recognized as a threat or danger to the body, and that causes allergies. There's another very bad side of the immune system that is sort of an overreaction to an infectious disease that results in septic shock. Septic shock actually is a very dangerous condition that kills almost 100,000 people in the US alone every year. And that is an overreaction to bacterial infections where the immune response starts to destroy the tissues and actually results in a shock and death, unfortunately. It's a major problem. Another example of this overreaction of the immune system happens after certain infections. For example, about 100 years ago, a flu epidemic called Spanish flu worldwide killed between 50 to 100 million people. And what was strange about this is that most of those people who died were young, healthy people that you would imagine they have a very strong immune response. And it turns out that now we know the reason why most of those people died is because they really did have too good of an immune response. Their immune cells secreted lots of proteins called cytokines in their lungs, and essentially their lungs collapsed trying to remove the virus. which we call cytokine storm. So those are examples of the bad side of the immune response that are very obvious, that cause disease and kill people. And then there's what I call the ugly side of the immune response. And the ugly side is not so obvious. It happens chronically, and there are many conditions that can cause that. What we classify it as chronic inflammation, but there are different types of inflammation. There's not just one type of inflammation. For example, during HIV infection, even if you treat the virus, in other words, if you suppress the virus to undetectable levels, the virus has caused so much damage to the immune system that these patients have what we call chronic inflammation. And when they reach their 50s or early 60s, they develop heart disease, cancers at a much higher rate for that period than healthy people. But other factors such as your diet, probably going to McDonald's too many times, or smoking and so on, can really cause what we call sterile immune responses. Sterile because there's not really an infection, but immune system is again experiencing a threat, a danger to the body, and responding to it in a chronic manner. We don't quite understand how this happens, but we think that this is an extraordinarily important condition, which actually leads to probably about 80% of the chronic illnesses, or it's directly or indirectly is associated with, for example, Alzheimer's disease, number of cancers, type 2 diabetes, even type 1 diabetes, cardiovascular diseases, autoimmune diseases I mentioned. These are all are result of this excessive inflammatory. They have different reasons why they happen, but this chronic inflammation slowly damages the tissues or blocks your arteries or destroys some of the structures in your brain that it cannot regenerate, that eventually causes neurodegenerative diseases and vascular disease, which are the major killers. So it has a really terrible cost, and we haven't really appreciated that until maybe a decade ago, that the inflammation, uncontrolled inflammation, that doesn't overtly cause disease, can be very detrimental for your health. And so we think that ME/CFS is part of this terrible health cost, and that probably is triggered for different reasons. From the reports of the patients, we think it's mostly an infectious trigger, but there are probably other triggers that cause the immune response to respond to it. But most people will recover from a flu, EBV, or bacterial infection. Somehow in ME/CFS patients, we think, at least that's our hypothesis, that the immune response sort of falls off the cliff and has difficult time in recovering to its normal steady state. And this is experienced by the patients throughout throughout their lives, for decades even. And at least that's our hypothesis, and I think there is good evidence being accumulated from our lab and others as well, that there is an involvement of the immune response. But the big question here is, why is it that this condition happens only in small part of the individuals who are having the same type of infection or same kind of environment? Of course, there's probably a genetic component, but we think that most of it is probably the environmental reasons that causes this. There are cases where a marathon runner, young individual, has an infection or goes to a trip somewhere in the world, comes back and is bedridden for years or decades. And that doesn't happen in everybody, and happens in some people. So we're trying to understand what is that trigger that sort of sustains this chronic state that results in a condition that almost in a vicious cycle feeds on itself. So our hypothesis is that one of the reasons, at least, could be what I mentioned earlier, is the microbiome. So let me tell you what microbiome is, why this is important, just like the immune system. Again, in the last decade or so, we began to appreciate there are trillions of bacteria that live in our gut, in our skin, in our mucosal tissues. And we didn't pay much attention to them before, but then it turns out that they have incredibly important functions in both helping us maintain our biology, importantly shaping our immune system, almost reprogramming, and also if it's not regulated in a proper way, causing lots of diseases. And in fact, there are more bacteria that live in our bodies than there are cells, and they are part of our ecosystem, and that we need them, but there's also a major cost of harboring these bacteria in our bodies. So this ecosystem has to have this equilibrium, and the equilibrium is maintained by different sets of bacteria. We very simplistically call them symbionts or regulatory bacteria. What that means is that these type of bacteria produce things that will try to suppress the immune response. So you have to remember that the immune system is trying to make a decision whether these bacteria can be a danger to the body or not. How can they discriminate between the pathogenic ones versus the ones that we need to keep in our bodies? And so the regulatory bacteria is sort of the piece, maintains this piece in potentially inflammatory bacteria. So there are those at the other side that can cause inflammation. Normally, they don't do that because, again, they are in this equilibrium in our gut niches or other areas in our body. And then there are commensal bacteria. Some of them metabolizes our food. They produce vitamins. They produce, again, different small molecules that our body needs, as well as acting on our immune system such that it shapes them to a point where the immune system can, or rather educates the immune system so that it can respond to infections better. There is evidence, for example, that if your microbiome is not set correctly when you're born or during your childhood, you're more likely to get allergic diseases. And there's even speculation the drastic increase in autism, for example, is due to our environmental changes that has resulted in different types of microbiota. So we think that it's extraordinarily important to maintain this balance, because if there's something that breaks this balance towards the inflammation side, and that could be because of an infection, an infectious pathogen that you're exposed to, or antibiotic treatment, long-term antibiotic treatment that will actually kill the beneficial bacteria as well in your gut. It could be your diet. Different types of food will give preference to certain types of bacteria that can establish in your gut. Many factors, and probably some that we don't even know, impacts this. And also it's important that how this microbiota was initially established in your body during birth, for example, whether you had breastfeeding, whether you were exposed to animal dirt that could have different types of bacteria, and so on and so forth. So there are many factors that decides what your microbiota is going to be like and what can actually trigger that. And if this happens, again, just like the immune system perturbation, it has a tremendous cost, most of which is actually through the immune system disruption as well. So when your microbiome is disrupted, that will cause some triggers to the immune system, cause inflammation. Again, there's evidence on the list you can see here is growing every day, anything from Alzheimer's to Parkinson's disease and cancers and so on and so forth. So it seems like it has really a profound impact on our biology and our health, and we're just beginning to appreciate this. So the big question is, does microbiome also is involved in ME/CFS? And this was one of our central hypotheses. And this was also based on some results that have been recently published that show that there were differences in the microbiota of ME/CFS patients. And we hear from the patients, for example, some case studies where they notice that things that make them feel better for their gut metabolism or their diet change, in some cases also helps with the disease symptoms. So there's a lot of anecdotal data, and it seemed like this was a very reasonable hypothesis as well, that something happens and that also disrupts the microbiota, which in turn results in disruption of the immune system. And this is sort of a cascade of events that feeds on itself, and the patients are not able to get back to their normal steady state. So that's our main premise. And using these two major hypotheses, we formed the basis of our research projects for this center, for the ME/CFS NIH Center. We are one of the 3 centers. The other one is in Columbia University. PI there is Ian Lipkin, and the third one is in Cornell. Maureen Hansen is leading that as well, and we are actually very, very closely collaborating with them. They are also interested in some of the similar questions, microbiome, as well as the immune response and other genetic factors as well. So the idea is that, as I mentioned earlier, if we could determine that there is a problem in the microbiota or the immune system of these patients, then maybe we can start to make some more directed hypotheses and look at the mechanisms. Why this is happening? And we think this is extremely important, not only for identifying biological biomarkers, but also potential targets that we can interfere with. For example, you can imagine if we find a certain type of bacteria that produces a certain type of metabolite that might not be a good thing, we might try to see if we could actually replace those bacteria with perhaps something like engineered probiotics. Or in other cases, there are metabolites that might be beneficial that are lacking in the patients. Maybe we want to replace those. Or if we see certain changes in the immune system, we might be able to target and fix that problem. So we think that this is a systems biology approach that will yield novel hypotheses and mechanistic understanding that hopefully will lead to more specific and targeted treatments. Again, as I mentioned, and I'm going to go in more detail about this, our focus is try to understand what we call the immune profile. There are dozens and dozens of different types of immune cells, different functionalities. We need to decode all of that. As I mentioned, there are trillions of bacteria that we need to sequence and figure out what kind of species that they belong to. And also we want to actually grow these bacteria out of the patients and determine what type of metabolites that they make, so that we can really go to the molecular mechanism using an integrative computational analysis, using machine learning and other approaches. And eventually we want to link that to the clinical data and hopefully translate it back to the patients. So it is a very comprehensive type of analysis. So let me start, go into more details about the immune analysis that we are doing, just to give you a little bit of a flavor on that. So the immune system, imagine it is our army in the body, and it's just like any army. It's very complex because it has to be ready for threats, enemies that it hasn't seen before. Roughly, we can divide our immune system into innate and adaptive immunity. What innate immunity is that these are the immediate responders. These are the cells that will start eating the bacteria as soon as you're exposed to them, or will trigger alarms that we are infected with viruses, or they will try to neutralize the toxins and so on. So these events happen within hours. So we absolutely need that. We cannot survive without that. However, we also need the adaptive immunity for 2 reasons. One is we want to be able to sustain the innate immunity. These are our, think of them as our generals or commanders that give, that tell these innate cells that this is what they need to do and so on. And in some cases, they themselves kill virally infected cells, for example, cytotoxic T cells. And of course, there are B cells that produce very specific antibodies that will go and bind to viruses and bacteria and and specifically allow them to be destroyed and neutralized, and hopefully eliminate every single pathogen from the body. Not in every cases, but in most cases. But the other very important aspect of the innate immunity, which happens in days and sometimes lasts up to a week to develop, is that it has a memory. Unlike the innate immunity, which will have to remember, it does not remember and will respond to a limited set of molecules that it considers as threat, the adaptive immunity is there in some cases for your life. For example, if you were exposed to measles or got vaccinated when you were a child, you develop certain type of T cells and B cells that will only attack the measles viral antigens or virus itself, and they will be maintained for decades in some cases. And that's the basis of vaccines, of course, that the adaptive immunity will remember the things that happen to you. And in some cases, there are certain type of viruses that you cannot get rid of out of your body. They just have to be constantly under check. For example, CMV or EBV, herpes viruses in general, they need to be under control all the time. That somehow we have to live with them, and it's really these T cells and the antibodies that make sure that they don't cause problems. And if you are immunodeficient for some reason, you lose your T cells or B cells, then those can be awakened and cause big problems. So of course, this is a very complex set of events because there are dozens of different types of infectious organisms, viruses, bacteria, pathogens, and not to mention that the immune system has to discriminate from the self so that it doesn't cause autoimmunity, all those bad situations that it might happen. So it has a built-in many regulatory aspects to it. So it has to control itself very, very tightly. And in order to do that, it has a huge division of labor. And my lab mostly is focused on a subset of immune cells called T cells. Again, these cells are sort of like the commanders or the generals or the regulators of the immune system. And this is just a very small set of different types of different types of T cells that you can generate. The names are not really that important. It can get very confusing sometimes, but the point here is that they have different functions. For example, this type of cell called Th1 is necessary for bacterial infections. If you are infected with Microbiome tuberculosis that causes TB, you better have Th1 cells, otherwise you're in big trouble. Others, like Th2 cells, are necessary to respond to parasites. They make certain type of what we call cytokines that are necessary to kill the parasites. But again, there's the dark side of these type of cells. Th1 cells can cause autoimmunity. Th2 cells can cause allergies and asthma if they're not controlled. And this type of cell that we study, Th17, also can cause severe autoimmunity or inflammation. And so that's why the immune system also has specialized type of cells we call regulatory cells. And their job is to make sure that these guys are doing just their job and they're not causing problems and destroying the tissue. So this balance between regulation and what we call effective functions is, again, going back to the first slide I showed, is critical to maintain that balance. And there are other types of cells that are very specialized. There are cells that kill viral infectious cells or tumors that are called cytotoxic cells. In fact, this is a big area of research in cancer treatment. We can actually arm these cells to kill the tumors. It's a very, very exciting field where we're also a bit involved in this to mobilize your cytotoxic cells to target the cancer cells, and in some cases we see an incredible responses, tumors being completely eliminated. So again, this is a very simplistic type of a classification. There are many subpopulations of each of these cells, and some of them we don't even understand why they exist, because your cells in the gut are going to be different than the cells in the lung versus the liver or versus the skin. The environment is also important. The context is also important. And we're trying to understand all this complexity by both studying the patient populations and the and trying to isolate these cells in vitro in the lab and decode their functionality. So how do we do that? How do we get into this complexity and try to identify, for example, what is Th1 cells, what is the Th2 cells, how can we work on that? So we have a great tool called Flow Cytometry. This is a sort of an essential machine for every immunologist. What it is that it's a machine that has a bunch of lasers, and these lasers will excite certain fluorescent molecules that can be detected by their detectors, and we can analyze that data using computer software. So, and we've taken advantage of this using specific antibodies, and antibodies are very specific. They will only bind to one molecule. So let's just say on a particular type of T cell, you have one molecule that will define that population. Then we can have an antibody that will lock into that, and you can have it conjugated to a fluorescent molecule, green, red, orange. We have many colors that we can use now. And then you pass through the cells. This is coming from your blood. As the cells are passing through, the laser is hitting on each of these cells, and if it detects a certain color coming from an antibody bound to that, it will be recorded as a particular signature that's shown as an example in this slide. So here we're looking at each of these single dots are one cell. So there's like 100,000 cells that we have collected here. And this is, here we're showing forward and side scatter, which means the size of the cell versus how granular it is. And you can see there are some large cells, very granular, and then there's some small cells. And so these small cells are what we call lymphocytes, and T cells are going to be within here. And so you say, okay, well, what type of cells do we have in here? You can put this region like a polygon and then look at this specific surface markers. For example, all T cells have a molecule called CD3. It's a molecule on their surface, and we have an antibody that will only bind to CD3, and that has a green color here. And so everything that is green is shown here. So these would be all the T cells in your blood, 86%. Whereas your B cells, another type of lymphocyte that make antibodies, have a very specific molecule called CD19. And so if you have an antibody that only binds to CD19 and you conjugate it to a red fluorescent molecule, everything is going to show up here. If there were cells that have both of them, they would show up here, that they would have both colors on them. Whereas those that don't have the CD19 and CD3 will show up here. They will show as non-T cell and non-B cells. So these would be NK cells and maybe dendritic cells and so on. Of course, we can go very, very deep in this. So now I showed you there are dozens of different types of T cells. So then we can say, okay, well, let's put an elliptical area here. These are all our T cells. And in this panel, we have other markers. Let's see what proportion of them are Th1, what proportion of them are memory, what proportion of them Th2. We have different markers for each of these populations. So we're able to do now 15, 20 colors in one tube, and we can identify, and we can do different panels. We're able to identify hundreds of different subpopulations. And we can also ask, for example, what type of molecules these cells are producing. We can trigger them, we can stimulate them as if there's a pathogen, there's an antigen, and these cells will start to produce their functional molecules, which are like their commands, and we can detect that. They're called cytokines, and that is extremely powerful. The one problem is not really a problem, but it's a challenge, is that everyone has a somewhat of a different profile. So in other words, not everyone has 86% T cells. Some people will have 70%, some people will have 87%, some people will have 60%. And that is just for the T cells. If you look at their, say, Th1 population, it can be 2% in one person, 5% in another, 20% in another. So there is a range, a huge range of different population in each individual, and that's your immune profile. And if you think about it, that makes sense, because all of us have been exposed to different type of pathogens, different environment. So our immune system is shaped, the type of cells that we have is based on our history, our historical exposure to immune populations, of course, our inflammation status and other factors, age, and so on as well. So this does make it rather challenging because we have to look at many individuals. Even in healthy individuals, we see sometimes tenfold differences in certain populations, and we're trying to decode that using various computational approaches. But the strength of that, actually, that's the caveat, but there's also an advantage of that, because it means that everyone has somewhat of a personalized immune system. And if we can link that to their other factors like the microbiome, metabolism, their clinical features, that could be extremely powerful to personalize even treatments or diagnosis towards that person. And this is not only for ME/CFS, but we've been doing this for other conditions, including cancer patients and breast cancer and cardiovascular diseases. And I mentioned HIV for many years as well. So I think it's a very exciting area, trying to really understand what does it mean to have a good immune system, bad immune system, ugly immune system, and what can we do to correct that. So that's the immune part. Similar type of complexity, maybe a different type of complexity also exists in the microbiome, because again, even though we all have a similar type of species of bacteria with very strange names that I have difficulty in pronouncing sometimes, but we all have somewhat different composition of these different species. And in fact, some individuals might have certain strains of bacteria that no one else has. Those might be rarer, but it's also definitely a possibility. And it's again a kind of personalized microbiota. Person 1 and 2 will have a different type of microbiota than each other, which again is set based on your environment. If you have used antibiotics and whether you had breastfeeding and so on and so forth. And that's another challenge that we have to grapple. So we try to sequence all of these bacteria and trying to create a database, but that alone would be too complicated to analyze. What we're also trying to do is to grow these bacteria from the patients and the healthy people and try to find those that are really, really important in acting on the immune system. We have developed assays that we can use these. So this is kind of shown here, the workflow that we do. We take stool samples, we sequence these bacteria, we assemble them, and we create a kind of a profile for every person. These are the type of bacteria that person has, and which we want to correlate with their immune response, immune system at the same time. But we have developed this condition called culturomics. In other words, we're growing individual bacteria, in some cases hundreds of them, from patients versus healthy individuals, and we're trying to determine what they produce, whether those bacteria, when they act on different immune populations, do they show a specific features. And that could be extraordinarily powerful, because if you identify specific bacteria strains, then we can do more biochemical analysis and hopefully identify exactly why that bacteria is causing the trouble that it's causing, or why it's actually good for you, identifying these metabolites, which I think would be very exciting as potential therapeutics. And the last point I want to make is the metabolism. So in fact, we're interested in the microbiota because of these metabolites that they produce. And what are metabolites? So we have our genome, which is sort of our blueprints to create the proteins through RNA, and these proteins will do lots of functions in our cells. But it turns out that we also produce lots of metabolites, or use lots of metabolites that come from our food, from our environment, from the microbes. When I say metabolites, for example, small molecules, things like glucose or lipids and amino acids, and these are not proteins, but they are small molecules that turn out to be extraordinarily important in shaping and programming our biology. And we think that many of these are actually made by different types of bacteria that live in us, that eventually act not only on the immune system, but probably other biological systems as well. So we want to be able to figure this out, and this is a major effort between other centers as well. There are hundreds of different metabolites in our blood that we're trying to understand and try to correlate again with the immune profiles and the microbiota of the same individual. And of course, ultimately, we want to bring all of this biology back to the patient. And in the patient, there's also complexity, and I'm not don't consider myself a clinician, but from what I learned from Cindy and other physicians, that this condition is not, this disease is not have a uniform symptoms. There are different levels of severity of fatigue, pain, sleep disorders, cognitive disorders, and so on, and different durations. So it's conceivable that there's a spectrum of biological reasons why these patients have this type of a symptom. So can we actually relate the immunological and metabolism and microbiota data to the specific clinical features or phenotype of the patients, so that will allow us to stratify the patients into groups that will enable us to better diagnose and better treat them. And we're using a computational approach, or developing computational approaches such as machine learning and artificial intelligence, to bring all this data together, and hopefully we'll have a greater picture in front of us. And that brings me to my last slide, scientific slide, that I want to quote Hippocrates. Hippocrates, the father of medicine, who lived about 2,000 years ago. What he said was, "It is far more important to know which person has a disease than what disease the person has." This is probably one of my favorite quotes. What Hippocrates was describing is something that we just rediscovered recently called personalized medicine. So normally medicine have developed, at least the modern medicine developed, as one size fits all. When I was in medical school, we were taught to treat diseases, diabetes, heart disease, brain disease, and so on. But it turns out that every disease has a complexity, that you can stratify patients based on variety of features, especially now using molecular markers into groups. And this has been extraordinarily successful, especially in cancer field. Now we're able to separate different types of lung cancer, for example, depending on type of mutations that they have, because each of these groups will respond to totally different types of treatments. And that starts to get into personalized. But it's still not personalized, because again, even in each group, you have different types of patients. So ultimately, what I would call the holy grail of precision or personalized medicine is really to personalize the medicine to the individual, to the one patient, that patient's biology, their clinical feature, their history, their environment, and so on, is what we need to focus on, and we need to treat the patient, not the disease in that individual. And I think that will be a major revolution in medicine that I'm hoping will happen in the near future, with the help of this extraordinary tools and data that we're generating, as well as the computational tools such as artificial intelligence. So I want to stop there and just sort of briefly mention also about our community outreach efforts, such as this webinar. We try to communicate with the patient groups on not only what we're doing, but also get their input on their disease. We have a website, kind of a blog, jaxmecfs.com, where we have spotlights on different advocates, scientists, physicians, and we also put educational blog posts about flow cytometry, microbiota, and so on, and I hope that's found useful by the community. You can follow me on Twitter. I try to post things of interest. We have a program manager, Courtney, who maintains our website, also has a Twitter account that you can follow. We have a separate community outreach program at Bateman Horn Center in Utah, where our clinical samples are recruited. You can contact them if you have questions. We have a separate community steering committee that we formed from physicians, advocates, patients that we plan to convene later this year. Obviously, we're working very closely with other centers and NIH on this center projects, which we've just begun some collaborations together. So I think this is very exciting for us as well, because we learn a lot from talking to the patients and understanding their complexity as well. Just very briefly, I want to tell you about the team. I am the program director of the center, and our associate program director is Julia O, who's also our microbiome expert. She's a young scientist here at Jackson Laboratory. I mentioned about our program manager, Courtney Gunter, our clinical core and community outreach team led by Cindy Bateman, Susan Vernon in Bateman Horn Center. We have Mary Dimmick. Some of you, I'm sure, know her from her advocacy work. Very excited to have Morris Papernick. He's a local ME/CFS physician, and we're trying to create a local clinic here in Connecticut, and so we could also actually recruit patients locally as well. Carol Barish, patient advocate, Patrick Mantucci, and Beth Mazur from ME Action, and we also work with Soul ME/CFS as well. Our immune microbiome team, we have a wonderful computational biologist, Peter Robinson here. I mentioned Julia O, our microbiome expert, as well as Mark Adams, who does our microbial genomics. We do most of the immunological studies. We have a metabolomics expert. We're also hoping to collaborate with other metabolomics core facilities in Columbia and perhaps in Cornell. Statistical support, Allison Matzinger in North Carolina. Again, we have a great bacterial chemist, which we hope will help us identify different metabolites that I just mentioned. We're also fortunate to have Paul Robson, who is an expert on single cell analysis and transcriptomics, and again, epigenetics, Duigo Uchar. So I'm going to stop here so that we have some time for questions, and I'll wait for the questions to come.
Thank you so much, Derya, and thank you so much for your team. This was a really great presentation, and we've already had some really interesting questions, so let's get started. The first question came in from Jim. How could an altered microbiome and leaky gut lead to mitochondrial dysfunction?
Mitochondrial dysfunction. So I don't know a whole lot about mitochondrial dysfunction, although we are very interested in that. I think Maureen Henson in Cornell, she would be the expert to better ask that question, but I can give sort of my opinion about it. Mitochondria, of course, is key. This is our energy plant in our cells, and it is very important for both the survival of the cells, but also it does regulate various biological aspects of the immune cells, and we're very interested in glucose and carbohydrate metabolism in that regard. So it's conceivable that the way your microbiome breaks down the food, the types of carbohydrates, will in turn impact your glucose or carbohydrate metabolism, which in turn might have an effect on the mitochondria. But I'm going to leave it at that because, again, don't have much expertise on it.
Great, thank you. Hopefully that answered your question, Jim. We have a question here from Carol. I believe you mentioned that dietary intervention has shown to improve the microbiome of patients with ME/CFS. Can you please comment on the nature of such a diet or provide a reference?
So I wanted to clarify that I didn't mean to say that it changes the microbiome and because of that there's an improvement. These are sort of the cases that I've heard that in some patients changing their diet or doing, for example, intermittent fasting or having a very low carb diet appears to improve these are anecdotal information, their symptoms, and it was a conjecture that these type of diets potentially could change the microbiota. It's something that we are very interested to explore but I'm not aware of a specific published study which have specifically looked at that, where there's a control study with certain diets, and then, you know, we know that certain diets will definitely change the microbiota, but whether the beneficial or in some cases adverse effects are because of that remains to be determined.
Okay, great, thank you. This is a question from Eric. The Cornell study showed ME microbiomes are remarkably similar to IBD patients. With IBD patients, they have little to no F, and I'm going to screw up this pronunciation, but little to no F. Prausnitzi. Are you finding this to be the case in ME?
So the microbiota, we're just beginning. So we've recruited about, I think, 20 patients so far, and we don't have data to answer that question from our lab. However, there's a paper published from Ian Lipkin's group, and I don't recall that particular species, if they have found that to be different. But it is an important point about the IBDs, or inflammatory bowel diseases. There seems to be some overlap in some patients. And again, we know that inflammatory bowel diseases have a very strong microbial component, which the inflammation is either caused by the microbial changes, or the inflammation in the gut actually results in this condition called leaky gut, which then changes the microbiota itself, sort of feeds on itself. So that is a very interesting aspect that we hope to look more carefully.
Okay, great, thank you. And then one question from Simus. I hope I've pronounced your name correctly. What is your opinion that the microbiome is not the primary problem, but the gut integrity, intestinal permeability disruption, possibly caused by a pathogen, which leads to inflammation, which in turn changes bacterial composition, and then the cycle is reinforced?
So can you repeat the very beginning? I kind of missed that. You were breaking up.
Sure. So would Dr. Unutmaz agree that it's possible that it isn't the microbiome that's the primary problem, but the gut integrity, intestinal permeability disruption, possibly caused by a pathogen, which then leads to inflammation, which in turn changes bacterial composition?
Sure. So I think we don't know a whole lot about it, but we don't necessarily need a change in the gut permeability, because these metabolites that I talked about, they can actually traverse through, even though there are lots of tight junctions in our intestinal lumen, especially from the colon. These things can pass through. You don't necessarily, because obviously that's how we get our breakdown food and other metabolites. If there is a disruption of the epithelium there, that I think is a very more severe condition that will most certainly will cause gastrointestinal problems, and perhaps cause things like inflammatory bowel diseases as well. But I agree. I think, you know, it's reasonable to think that something is going on there, and that any type of infection perturbs that balance between equilibrium, let's say, that is maintained in our body. So you have to realize that when you look at this microbiota, we kind of know who are the bad guys and who are the good guys. And in many healthy people, you find these different types of bacteria. You have the good ones and you have the bad ones, but they're not necessarily causing any trouble because we have that equilibrium. So all it takes is some sort of an infection, some sort of a perturbation, but again, it doesn't have to be an infection itself, can disrupt that balance towards more pathogenic level and result in trouble.
Great, thank you. Question from Diane. What is the function of the complement innate immunity cells, and how does it interact with the microbiome, T cells, or autoimmunity?
So complement is part of the innate immunity. What complement does is that it usually works together with antibodies, and it will literally cause killing of the bacteria. So it will punch holes on the membranes of the bacteria. It has other immunologically regulatory functions as well. So complement is very important in bacterial infections. It's a very good question. What is the role of complement in the microbiota? Because these are also bacteria. There's a specific type of antibody called IgA that's actually secreted into the lumen of the gut that seems to be specially evolved to regulate our microbiome-related bacteria. And the role of complement there is still unclear, but I'm pretty sure that it has a very important function in sort of filtering out the potentially pathogenic bacteria that live there together with this IgA antibodies. But I don't know anything specific to ME/CFS and complement changes.
Okay. A question, quite an open-ended question from Aaron. Are there any interesting findings emerging from your current patient studies?
Yes, we're starting to see quite interesting differences in the immune system. We're still doing a lot of analysis on that and hope to start writing a manuscript on it very soon. So I think we initially focused on the immune profiles, and there's clearly definite perturbations in different types of cells that we're still trying to sort through, and hope to be able to report that soon.
Okay, great. And then last question from Larry. Do you see evidence of microbiome dysbiosis gliopathy in ME/CFS?
We haven't looked at that. Again, there's fairly limited studies. I'm going to refer to Lipkin's study and a couple other microbiota studies, but we don't have anything specific to answer that.
Okay. All right, so we're coming up on to the top of the hour. We do have a few more questions, but perhaps I'll forward them over by email and we can get responses out to those folks who we haven't had time to talk about today. So sure. With that, I'll hand it over to Emily, who's just going to review some upcoming webinars and where you can go to get more information.
Thank you.
Thank you so much, Dr. Unutmaz, and thank you, Sadie. We really appreciate all of your expertise and sharing your knowledge and updates about your work with our community. Everyone, thank you so much for joining us at our July webinar in the Solve ME/CFS Initiative webinar series. Please follow us on Twitter and of course Facebook, where you can join the conversation and provide feedback about these webinars and the information we provide. Additionally, as you know, this webinar was recorded and will be available on our YouTube channel at Solve CFS on www.youtube.com. If you have any questions about future webinars, you can always go to our website at solvecfs.org/webinar. Join our 2 monthly e-newsletters at solvecfs.org/newsletters. And of course, if you are interested in becoming a part of our biobanker patient registry, you can see that on the solvecfs.org/biobank. And of course, everything we do is funded by patients and donors just like you. So if you enjoyed this webinar and you enjoyed the information we provided, we do encourage you to help fund our progress and fund the work that we do here with a donation at solvecfs.org/donate. Thank you again for all of your time. We'll do our best to get answers out to the questions that we were unable to get to on the webinar, and have a wonderful rest of your afternoon or morning, depending on which side of the country you're on. And we'll see you again at the next webinar. Have a wonderful day.